So I'll be giving an introduction to high-throughput sequence data and understanding the origin and shape of the data. And what I mean by the origin is just where we're starting from, and the shape of the data just in terms of like what file formats we're going to end up with and how the data is structured. So in general, we want to move from some biological sample represented by these test tubes to a list of variants. And so we have a multi-step process where we're going to prepare our biological samples, then we're going to sequence them. That's going to provide us with some pile of reads in a number of files. We're going to take these files and put them through the GATK best practices, and that'll produce a list of variants, which is usually the thing that's of interest to us. So the first thing that we have to do is what we call library prep. And this is where we take our biological sample, in this case, some individual, and we're going to extract either DNA or RNA from this individual. And because sequencers can only, at least Illumina sequencers, can only sequence DNA, if we take RNA out, we have to convert it into DNA through a process called reverse transcriptase PCR. But once we have our DNA, we can go through a process that we simply label as purifying fragments, but this is going to be very dependent on what your application is. So this will often involve like some amount of PCR, maybe some sort of hybrid selection. It could also involve size selection of your fragments. So once you have your collection of fragments, you can attach adapters to these DNA fragments. And so you have these double-stranded DNA fragments that now get these adapters that are ligated to both ends of the DNA. And the adapters are also made out of DNA. You can, depending on the concentration of your DNA, you may need to PCR it up so that you can increase your concentration. And then we call this a library. Once we have built our library, we want to sequence it. And so we have our DNA libraries in these test tubes. We now need to put them on this flow cell. A flow cell is an interesting device. It's about it looks a bit like a microscope slide. Some of them are about the size of a microscope slide, and while others are about this big, a little bit bigger. The flow cell is a device that has these ports where you put the contents of your DNA library on, and it flows across this lane, and the bottom surface of the flow cell is chemically modified so that the DNA will bind to the bottom of the surface in such a way that there are patches that many different copies of that same molecule, like you'll get a single molecule that will land on one of these patches that will then amplify that particular strand of DNA so that a single patch will now have many copies of that same strand of DNA. And you have roughly 1 billion to 10 billion different patches on the entire flow cell. And once you have these patches of DNA, we flow nucleotides across this flow cell in such a way that base pairs are added one base at a time, and they're added with a dye, so that these patches each now have a color depending on what DNA base is on the sequence that's attached to the bottom of the flow cell. And so when you image it at each step, you can see each one of these patches has a particular color, and each color corresponds to a particular base. And then this will result in what we call an enormous pile of short reads. And by enormous, we typically mean for each flow cell is going to be about 10 or 1 billion to a billion, or 10 billion reads. In the case of the NovaSeq, as many as 20 billion. The raw sequences are typically provided in a format called FASTQ. FASTQ is just a text format. You can read it with a text editor, like Sublime, but these are typically very large files, so that it's not recommended that you open them with Sublime. The format is of a single read. Will start with this @ character, and then the name of your read. The name of the read will oftentimes contain information like the name of the barcode of the particular flow cell. It may include information about which lane of the flow cell it was sequenced on, and it may even provide some sort of XY coordinate about where exactly on the lane that particular read was sequenced. The content of the read is, of course, here. And then after this, you can get this series of characters where each character represents the quality of the base. And so whenever the sequencer makes a base call, it also makes a guess as to how confident it is. Each one of these characters represents some Phred scale quantity. And a Phred scale quantity is just a log quantity of the probability of an error. It's a nice scale, so a Phred scale of 10 means that you're 90% confident. A Phred scale of 20 means that you're 99% confident. And with each increase of 10 in the Phred scale, you're adding another 9 to your confidence. And here we just have a little plot of the accuracies and errors for each Phred scale. And so generally, when you do raw sequencing on an Illumina sequencer, we consider Q20s, so things that are 99% confident, to be a good base call. Something that would be, a Q30 would be very good, and a Q40 would be great. And we do see that range. It's also important to know whether or not you're going to be sequencing, say, whole genome or exome. The different types of data have different features that are important to keep in mind when you're doing your analysis. So here, this is just a reference that we have some intergenic region, some exons, and an intron between our exons. And here, each red line represents a read that has come off the sequencer. The red lines that have a star in them represent reads that have a difference at that particular site. And these differences here are indicative of a variant. In practice, this is what this looks like in IGV. So each read is one of these gray rectangles with a little arrow at the end. The gray rectangle indicates that it matches the reference that is here on the bottom. We're zoomed fairly far out here, so we can only see colors. Each color represents a different base. And because we're zoomed out so far, we can't actually write each. There's not enough room to print like an A, T, C, or G. Here we see a coverage track, and this coverage track has a maximum value of 58. And so you can see that there's some difference, maybe almost of a factor of 2 across this whole region. We can't see the entire depth here because this is a screenshot, and you would be able to, within IGV, you would be able to scroll down to see all of these reads. There are a couple key features to notice. One is that each one of these reads has a direction, and that's because each read is sequenced from 5' to 3'. And depending on which strand you grabbed of the double-stranded molecule, depends on which direction it's going to map to the reference. Many of these reads also have some differences that are represented by various colors, and these are differences from the reference. Most of these differences are sequencing errors, and so we believe from experience that things like this here and here are likely due to some sort of failure of the sequencer to guess the correct base. But there are other differences, like here, that from experience lead to us to believe that this is an actual biological variant. Here, we're looking at a human sample, and you'll notice that about half of these reads have this difference. And the reason why that's important is because humans are diploid, and so half the reads came from one chromosome and half the other chromosome, and it turns out that one of these chromosomes had the difference in it. We can't label each read as having come from a particular chromosome, and so there's no sorted order here. We don't know that all these came from maybe the paternal haplotype or the maternal haplotype, but so these may come in any order. And you'll notice that there's another variant down here, and you'll notice that they're in a different order from these. Likewise, there's yet another variant. This here is an insertion, and this insertion means that there are extra bases that don't match the reference. And within IGV, if you want to look at those extra bases, you can hover over this eye. Another feature that's important to look at is this coverage track. Between whole genome and exome, you'll notice that the coverage tracks vary widely. Here is the targeted region within the exome, and we get very high depths, over 100, in the regions where we're targeted, but there are some flanking regions where we get lower depths over nearby but non-targeted regions. Even though even though we do see evidence of a variant here, we'd never be able to call it because we wouldn't be able to distinguish between this being a biological variant or some sort of sequencing artifact. Yes?
That was a question there.
For distinguishing between sequencing errors and variants, is that just kind of a matter of looking at the proportion of reads that a change shows up in and kind of throwing out the ones that only appear in a very small fraction of the reads that you're looking at in your sample?
So in general, yes. So there are a number of statistical tests that are run when in the GATK, which you'll learn later. And these statistical tests basically look to make sure that they look to see, what is the probability of, sort of roughly speaking, what is the probability that this was caused by sequencing errors or biological variant? And if the evidence strongly suggests a biological variant, then the caller will make a variant call.
With regard to the exome sequencing, why is it that you do get any reads at all in the intronic region?
In the intronic? Why do they not have a sharp boundary? Yeah. So there are some exome capture, some targeted PCR methods that do have sharp boundaries. One of them is called amplicon sequencing. But there is another process called hybrid selection, which is what this is. And hybrid selection, you design baits that are complementary to this region of interest. They don't have to grab that region perfectly. they can grab a region that's nearby and get something that's close enough. And so they bind less efficiently, which is why you see fewer of them, but you still will get some. Depending on what kit you use, we just described this one as kit A and kit B. You may get two wildly different coverages over slightly different regions. So for example, this kit, for whatever reason, appears to target this intronic region, while this kit appears to target the exonic region. And so depending on your application, you may want to prefer kit A over kit B. Quality control is essential. When sequencing is expensive, and so you want to catch your errors as early as possible. There are a lot of common problems. One is that you didn't do enough coverage, or you didn't do enough sequencing. Another problem is that you may end up with a high proportion of chimerism. So in Illumina sequencing, these are paired end sequences. So you end up with each fragment has has two reads. And one read may map to one part of the genome, while another read may map to a very disparate part of the genome. And we call these chimeras. And they're not always indicative of a problem, because they do occur in structural variants. But there are sometimes library prep, things that can go wrong during library prep that can result in a high proportion of chimerism. Another problem that we sometimes see is like a strange insert size distribution. Having one that's too large or too small can indicate that something went wrong during library prep. Also, additional peaks in places where you don't expect them can also indicate various problems. When we do shearing, so that we can get fragments of the appropriate size, the shearing is a relatively violent process where acoustic waves are focused onto the DNA, and these acoustic waves can actually cause some of the bases to oxidize. And when that happens, that can result in either subsequent stages of PCR to go wrong, or if these oxidized bases show up on the sequencer, that can also produce incorrect results. Another problem that you might have is that you may have a library size that's too small. This could be a result of, this is usually the result of having a concentration of DNA in your sample that was too low. And you can do a lot of sequencing, but if you do a lot of sequencing with a library size that's too small, many of your molecules will be duplicates of each other. And those duplicates don't provide any additional information about the biology. And so you're spending a lot of money unnecessarily. So two common differences that we see between, let's say, exomes and genomes is that with an exome, there's a very significant unevenness in the distribution of coverage. One metric that we use for measuring how uneven coverage is something called fold 80. Fold 80 is just the measure of how much extra sequencing we would have to do to bring up the average target to the mean target coverage of that particular target. Another thing that can happen is that you can get reference bias. The baits are designed typically to capture the reference. If there is a variant in your DNA, the baits will bind less efficiently to that DNA, and so you can end up getting a larger number of reads that have the reference base than you expect by chance. With whole genomes, sometimes you will get a much higher proportion of unmapped reads. That's okay. Sometimes, when you're sequencing everything, maybe not everything is going to align well to the human genome. That could be because you may have bacteria in your sample or other contaminants. And it can also be because there are other human contigs that are just not represented in the reference. And another problem that we sometimes see is that we can get a high percentage of adapter in our reads. There are a number of error modes that are common and that can interfere with our high-quality data generation. One common problem is regions of the genome that are high in GC content. GC-rich areas are problematic because these GC base pairs have 3 hydrogen bonds, and that rather than 2, which the AT base pairs have. And that results in these molecules with high GC content having a higher melting temperature. And so sometimes they don't always dissociate from each other. And so they can be more difficult to sequence. And so here you can see that in regions of the genome that have lower GC content, we see a decent number of reads, but with a particular kit, we lost a lot of reads in this high GC content region. The distribution of your coverage matters. If your different kits will have different coverage distributions, you may have, and just depending on what your application is, one kit may be preferable to another. So if you have uneven coverage, one thing that can happen too is it can inflate your, it can give you, if you have a high depth in an uneven territory, it can give you a nice mean coverage. And so sometimes looking at just mean coverage is not enough. You want to look at other metrics that indicate, like the fold 80 metric, that indicate how even your targets are, or how evenly your sample is covered. We even see unevenness in coverage between read groups. I think this is a little bit less common than what we see in between, say, exome and genome. But different read groups, for various reasons, may have different coverage. And sometimes they don't have enough coverage. So sometimes we want to sequence additional read groups so that we can combine the two results to increase our coverage if the first run wasn't enough.
Why did you say the read group 2 is problematic in the previous slide? It's just so you would not use the...
Well, I think that's very dependent on your application. I think that it's actually okay to use to add this because it may have failed in the sense that it has very low coverage, but it can be, I think in generally it can be added if it were being used as a top-up.
So how do you define a read group? Is it like a replicate of a sample you ran in the different lane or in the same lane again? Or?
It would be in a different lane. Sometimes we also see high percentages of chimerism. Usually when you see reads that have bright colors in IGV like this, that's an indication of some sort of problem. Each one of these colors means something different, depending on the particular options that you have set in IGV. And so you can't always remember what these colors correspond to. And so in this particular case, the colors indicate that the reads are mapped to a different contig. And that's a indication of various problems that could have occurred during library prep. But it's also something that you can see when it's not uncommon to see chimeras in regions of structural variance. But when you can get your chimeric rate down, so if you have a really high concentration of chimeras, when you your other metrics are affected. So for example, here we have a high amount of chimeras and a relatively low percent selected bases. And so if we can reduce our chimeric rate, that'll increase our percent selected, and it can also increase our percent of targeted bases at 20x. Yes?
So if it's considered chimeric because it maps to a different location in the reference, why does IGV show it here? Because it's part of the pair?
No, yeah, because it's part of a pair. So so this read might be read 1, while read 2 may be really far away, either on the same chromosome or on another chromosome. It's also important to look at insert size distributions. It's important, depending on the particular protocols that you're using, you may have multiple peaks, but it's important to know when you have a peak that's abnormal. As here, we noticed a very abnormal peak in some data that was sequenced with immune-compromised individuals, where the samples were taken from saliva, and these saliva samples had very high concentrations of bacteria. And the bacteria had enough similarities to the human reference in places that they managed to align to the human reference, creating these gigantic coverage peaks at various places. These rainbow-like areas are regions where the read doesn't match the reference. And what's happening is we call these soft clip bases, and these are regions where the aligner has given up and says, I don't know, something funny happened. But these reads are all, most of these reads here are bad. And you can notice that through here. So just to recap, we're going from a biological sample all the way through sequencing and processing to generate a list of variants.